Yes. Welcome. Good afternoon. Welcome for the second part of our learning. Second part of our learning, our studies. We are going to do science lab. And then as days go by, we are going to switch. We are going to add more subjects that we are supposed to be covering. So it's not going to be just maths and sciences, but we are also going to see about English and other subjects. We are going to see about English, English reading classes, reading, and also ESL, English as a second language, ESL, English as a second language. So, but now let us look at science. Welcome to Science Lab. And wow, remember we are dealing with science, heredity and society. This is our topic. Yesterday we looked at physics, heredity and society. Heredity and society is the cause. Heredity and society is the cause. This is a subject on its own. This is a subject of its own, and it's a division of biology. Remember, it's a division of biology, and as I told you before, it is a discipline of biology. Remember, we've been talking about DNA, and we're talking about cells. We're talking about cells, and we've been talking about nucleus and all that good stuff. Nucleus, remember we said that nucleus plays a very essential part in the cell. The nucleus plays a very essential part in the cell because it acts as a controlling center. Without the nucleus, the cell may not survive. Without the nucleus, the cell may not survive in the living organism. The cell may not survive in the living organism. So, nucleus is a control center of the cell. So now we're going to look a little bit more about DNA. DNA is a large topic. But before we look at DNA, remember we were talking about some problems that are associated with DNA. Some problems. There are some sicknesses that are associated with defects of the DNA, the genetic makeup. So we mentioned just but a few. The other time, remember, we talked about the Tay-Sachs disease. And remember now, and we also talked about schizophrenia. We also talked about schizophrenia as one of those diseases, and that we can go through in our whenever there's a defective cell, whenever there's a defective cells in our bodies. So now we also have some other sicknesses that we need to know about, some other diseases that we need to know about, and also how they are, whether they are cured, or how they can be prevented, or how they can be treated. How they are cured, how they can be prevented, or how they can be treated. So let us learn and know about these problems, So now, before another fact that we need to know about the genes or the genetic makeup is that there are genetic materials. There are genetic materials that are present, that are always present in our DNA makeup. There are genetic materials that are always present in our DNA makeup. Now, study of DNA is not very old. DNA is not very old. It's a new kind of study. The study of DNA is a new kind of study. It's just a few years old. It's a new invention. It's a new invention because before the innovation of DNA studies, little was known about DNA apart from the genetic material. Little was known about DNA apart from the genetic material. But as science has continued to evolve, now people have learned more. Now scientists have discovered more they've discovered more beyond just genetic makeup, beyond just the genetic material. So it's good to know that prior to the 20th century, not much was known about genetic material. So that just tells us that the study of DNA has only been known mostly in the 20th century. The study of DNA has only been known mostly in the 20th century, and therefore little was known prior to the 20th century. Not much was known about genetic material. And so what was known, the only thing that was known was that the genetic materials was able to store information. So the only thing that was known was that the genetic materials was able to store information. The only thing that was known was that genetic materials were able to store information. They were able to store information. So now about the diseases, and some pathogens that can affect us. One is called a bacterium, and streptococcus, and then there's bacterium, streptococcus pneumonia, bacterium, streptococcus pneumonia was implicated as an etiology. It was indicated as etiologic. That means the study of diseases. The study of diseases, etiologic. So that a bacterium thing came out. A bacterium thing was discovered. Is a kind of a bacterium. That's kind of a bacteria or something that can affect us, that can affect the DNA, and so it's a bacterium. Is a bacterium, streptococcus. It causes pneumonia, and that was implicated as etiologic. And then now we have problems such as macrophages, and now something macrophages is that which help us to prevent, to help us fight diseases. A macrophage is on the first line of defense. Is on the first line of defense, and it ingests foreign objects. It ingests foreign objects via a process called phagocytis. Macrophages are our first line of defense. Whenever there's an attack in our DNA or within our bodies, then a macrophage is our first line of defense. It is our first line of defense because it ingests the foreign object via a process called phagocytosis. Now we have a virulence factor, and then a virulence factor, that is a factor that allows pathogen to colonize the body to cause the disease. A virulence factor is a factor that allows the pathogens to colonize the body and cause the disease. So now, whenever your body has been diseased, it's because there's a factor called a virulence factor. It's a virulence factor that causes our bodies to allow pathogens. So pathogens are germs. Pathogens are disease-causing germs. Pathogens, when they talk about pathogens, it simply means germs or something that causes diseases. So germs are also known as pathogens. Germs and anything that causes us to be sick are known as pathogen, and they colonize the body. They take charge of the body, and then that causes disease. That causes the body to suffer diseases. So that thing which causes the body to suffer disease is called a virulence factor. Now let us look for treatment. Okay? So now one of the most common ways of treatment, one of the most common ways of to get treatment and is to use a process of breaking up the cell to treat with enzyme. You break the cell to treat with enzyme. So if the cell has been attacked, so to get treatment is to have it broken up. You have to break open the cell and treat with enzyme. You have to break open the cell and treat with enzyme to destroy protein and to the rough cells. So you destroy the proteins and to the rough cells. You destroy proteins and then you add to the rough cells. You destroy proteins and then you add to the rough cells. So that's the treatment. That's the treatment. So I don't know how that, the doctors know best how to do that, but because how that means it's some sort of surgery or something, because they have to break the cell. And then they break open the cell, and then they treat it with an enzyme. So an enzyme is something that activates it activates. So they put some medicine in there to activate the cell, and then they destroy the proteins that are in there, and then they add rough cells. Okay? Because once there's a defect in the cell, so they have to treat it by breaking it open, put some enzyme in there to reactivate it, and then they add rough cells, Sound like a process of purification of the blood or something, but the doctors knows best what when they talk about breaking open the cell. So the result is a transformation. The result, when that has happened, then they get transformation, which is rough cells become smooth. They have capsules. So once that treatment has been done, then a transformation comes through. Now you have new cells. Transformation comes through. Now you have new cells. That means the rough cells become smooth. And now you have new capsules. Now I love that part because it makes me feel good. It's like a transformation for those who are Christians. Now you become a new creature. So when they treat you like that, your cells get some relief, and then now you are transformed, and now the rough cells become smooth. It's like Prophet Isaiah saying, "Prepare ye the way, make the rough roads smooth." But now, back to what I was saying, the result is transformation, whereby the rough cells become smooth. And then they have new capsules. Then, apart from that, there's also pneumonia vaccine. Pneumonia vaccine is a form that can be used for treatment. Pneumonia vaccine. So, pneumonia produces about 90 different capsules. So there's a vaccine called S pneumonia. It produces about 90 different types of capsules, and had to create a single vaccine to that many antigens. Now. So now the antigens, the things that causes diseases in our bodies, they are so many. So now the vaccine that we have carries up to 90 different capsules. The vaccine that we have, especially for pneumonia, carries up to 90 different capsules. Remember, we are just talking about types of treatments and the results we get. And the results we get from doing that. So now, apart from that, there's also a disease called sickle cell. Sickle cell, just roughly saying, is a disease that attacks the cell, the red blood cells. Sickle cell is a disease that attacks the red blood cells, and it attacks their formation, and it gives them a shape that looks like a sickle. Hence the name sickle. A sickle is that thing that farmers use to cut grass with. So sickle cell is a DNA problem, and it attacks the red blood cells. Sickle cell is a DNA disease that attacks the red blood cells, and it can be passed through. A parent to a child. depending on your genetic makeup. If you come from a genetic, if you come from, it can come from your paternal side of the family, or from your maternal side of your family. It can run from your family, in your family lineage. So it depends. So now, in some cases, you can be a carrier of sickle cell disease. You can be a carrier of sickle cell disease, but it cannot hurt you unless your parent are both sickle cell positive. Then now that's where the problem is. So if only one parent is sickle cell and the other one has no sickle cell, so if the child is born with sickle cell, he or she may survive, but then now if later in life, so now that's where testing is important now. Later in life, that child will need to make sure that when it comes a time to marry, he or she is going to marry a person who does not have the sickle cell trait. So if you have sickle cell trait, it may not be harmful unless both your parents had it. If only one parent had it, you can have it in your system, but it may not affect you. You may not feel any pain or any signs of sickness. But then the problem comes if both parents had it. If both of your parents had it, then definitely there's a chance that you're going to have sickle cell disease. Now, sickle cell disease is a blood that attacks the red blood cells in our body. It makes them weak, and it reshapes them. It reshapes the red blood cells in our body by turning them into sickle cell shape. And then it causes a lot of pain. For the offspring. It causes a lot of pain to the offspring. So these are some of the diseases of genetic diseases that comes have been discovered through DNA and or through through blood test. So now, moving on. So that's some of the diseases that we do have that are out there. People can survive through them because there are some treatments now, but now research is still going on, especially for sickle cell disease. Research is still going on. So now I want us to learn, unless I forgot any other kind of disease out there, but I want us to learn about chemical subunits of DNA. So now DNA has some chemical subunits that we need to know. DNA has some chemical subunits that we need to know. And so organisms contain two types of nucleic acids. Organisms contains two types of nucleic acid, and one of them is nucleic acid. So one of them is called deoxynucleic acid. So that's the main DNA that we are talking about, deoxynucleic acid. And then we have the ribonucleotide or RNA, ribonucleotide or RNA. Then then now they are both nucleotides. All these subunits of DNA are all nucleotides. Together they are called nucleotides. Together they are called nucleotides. Now they are components of nucleotides. They have components, whatever is in there, they are components of nucleotides. One of them is called nitrogen. So nitrogen containing base. So nitrogen containing base, and then they have purines, adenine, so nitrogen containing base, and then we have purine adenines, and then now we also have something. So purine adenine is or a purine adenine in bracket A for adenine, and then we have guanine in bracket G for guanine, and then so it's guanine pyrimidase. So those are the components of the subunits of DNA. Those are the components of the subunits of the DNA, or components of nucleotides. Remember, together they are called nucleotides. So nitrogen containing base, purine adenine or A, and guanine G, pyramids. Guanine or G, pyramids. Guanine or G, pyramids. So the genetic information is stored. Genetic information is stored in sequence of DNA. Genetic information is stored in sequence of DNA. And remember, DNA is a sequence. Is a long chain. It's like a long string. It's not just a small thing. Where when they talk of DNA, it's a long chain. It's a long chain. It's something long. It's a sequence. It can go up to together. DNA carried information. So the sequence of DNA, they are clustered as chromosomes. The systems of DNAs are clustered as chromosomes. They are clustered as chromosomes, and they carry genetic information. Okay, so DNA in total, DNA in human chromosomes, they are up to 46 in human DNA, or whatever the chromosomes. The sequence in DNA are called chromosomes. The sequence in DNA are called chromosomes. And then in human being, so this is the thing to note, in human being there are a total of 46. So that means there is a 23 X chromosomes, and then there's a 23 Y chromosomes. So together they make a total of 46 chromosomes, a total of 46 chromosomes, and they go in a sequence. So one, they go up to 2 feet. They go up to 2 feet, which means it's 1.8 meters. 2 feet or 1.8 meters. So remember the metrics. We were talking about the metrics. So when we talk of 2 feet, we mean it translates into 1.8 meters long. So a DNA, the sequence of DNA can go up to 1.8 meters long, can go up to 1.8 meters long. So, and remember they are folded, so they are folded, otherwise if they were not folded, people will be working with their DNA, following them, okay? So our DNAs are folded, they are compacted into our bodies. They are folded, they are compacted into our bodies. You can't see them, but they are folded into our bodies. They are in our genetic makeups. They are in our genetic makeups. And now they are folded and compacted by a factor of up to 10,000. Take a minute to digest that. Take a minute to digest that. They are folded and compacted up to over 10,000. So we have over 10,000 compacted DNA in each one of us. We have over 10,000 compacted DNA. They are folded up, compacted up to over 10,000, and within that compartment there are micromolecules. Within that compartment there are not micro but macro. There are macromolecules which contains and proteins, carbohydrates, lipids, nucleotide acids. Proteins, carbohydrates, lipids, and nucleotide acids. So we have some acid in us called nucleotides, and we have lipids, and we have carbohydrates, and we have proteins, So the best definition of chromosome, you want to write it down. The best definition of chromosome is that chromosomes are carried genetic information or DNA, and they are found in the nucleus, okay? Chromosomes are carried genetic information or DNA, and they are found in the nucleus. They are found in the nucleus. So we're gonna look at the components of nucleotides. Components of nucleotides, which remember I mentioned a little bit. I'll mention a little bit down here. One of them was nitrogen containing base, and they are purines, adenine, and they are guanine pyramids, So now let us look at how to identify what is nitrogen, what is purine, what is pyramids, and then we can stop there for today. So nitrogen pyramid, basically are okay. So the purines, adenine, and guanine, they contain the nitrogen. So they have a base containing nitrogen. So that is the most characteristics of them. So the purines, adenine, and the guanine pyramids are the components of the nucleotides, and they contain nitrogen. Nitrogen is a chemical so that are contained into the purines, adenine, and guanine. Then we have the pyrimidines. Pyrimidines are cytosinous. Pyrimidines are cytosinous. That sounds like a hormone or something. Cytosinous, and for Let me erase the board because we want to make sure we understand this. So we take some notes, so I write it down for you to remember. It's gonna help you in your quiz. It's gonna help you in your quiz. So I want you to have fun with this as you learn, so you make your classes easier. Have fun with this as you learn, so it make your classes easier. So now, components of nucleotides. So components of nucleotides. Components of nucleotides. Okay. Components of nucleotides. So number one component is called purines. Purines. Adenines. So number one is purines. Purines adenine. Okay. Purines adenine. It's represented by the capital A, letter capital A, and then they go together. Okay. So purines, and so we have under purines we have adenine and guanine, adenine and guanine, adenine or guanine, and that is the guanine is represented by the letter G. Guanine is represented by the letter G. So besides purines, we have pyrimidines. Besides purines, we have pyrimidines. Besides purines, we have pyrimidines. Okay, and pyrimidines are components such as cytosines. So cytosine, or let us see, cytosine, I believe is Y. Yeah, cytosine, or let us see, is one of the pyrimidines. And then we have thiamine T. We have thiamine. We have thymine, or letter T. And then beside thiamine, we have uracil. Beside thiamine, we have uracil. Which is represented by letter U. You have uracil, which is represented by letter U. Okay. Then now we have DNA. We have DNA. We also have to know the components of DNA. Okay. So now let me erase the numbers, because if I put the numbers, then the DNA don't want you to confuse DNA as part of the nucleotides. Okay. Now we've done with the purines and the pyrimidines. Now we are dealing with the DNA as a component. DNA is a component of its own. So now we have DNA. Now we have DNA as a component of its own, and now it is represented as so this is where the letters comes in. This is where the letters come in. So we have A, G, T, and C. So we have A, G, T, and C. That means DNA contains both adenine, guanine, thymine, and what's the last one? So at least we yet to find out what the C is for, but at least it has AGTC. DNA has AGTC. DNA has AGTC, and then RNA has AGU and C. So RNA has AUG and C. AUG and C. AUG and C. AGU and C, sorry. AGU and C. Yeah, you have to write them in the correct order. So it is after A, we have G, U, and then we have C, and then we have C. So AGU and C, AGU and C are under the ribonucleic acid. AGU and C are under the ribonucleic acid, whereas AGTC are under the dinonucleic acid, or DNA in short, dinonucleic acid or DNA in short. Then we have sugar. Which is also called ribose. So we have sugar. Or another name for sugar, the scientific name for sugar. When we talk about for DNA purposes is under what we have under sugar. So sugar is just sugar is part of the components. Sugar is part of the component, and now under sugar we have something called ribose. Under sugar we have something called ribose. Okay, so sugar is just sugar, or you can call it glucose, but sugar is just sugar. But under sugar we have ribose or deoxyribose. So we have ribose nucleic acid, ribose RA, or deoxyribose. Okay, deoxyribose, and this is DRA. Deoxyribose, or not DNA. Deoxyribose DNA. Deoxyribose DNA. So it's a long one. So you have to say it all together. Deoxyribose DNA. So those are the components of the nucleotides. Those are the components of the nucleotides. Those are the components of the nucleotides that are found in the DNA. Those are components of nucleotides that are found in the DNA, and we've learned that they are nitrogen containing base. They all contain nitrogen. They all contain nitrogens, and we've listed quite a number of them. The purines. Under purines, we have adenine. Under purines, we have adenine, represented by the letter A. Remember, the letter A has to be in capital. And then we have the guanine. We have guanine represented by the letter G, capital G. And then we have the pyramids, pyrimidines, not pyramids, but pyrimidines. And under pyrimidines, we have the cytosina. Cytosina is represented by the capital C. Cytosina is represented by the capital C. And then we have thiamine. Thiamine is represented by the letter capital T. Thiamine is represented by the letter capital T. And now we have uracil. And uracil is represented by the letter capital U. And then now we have DNA as a component of nucleotides itself. So DNA itself is a component of the nucleotides. And now under DNA we have letters, all capital letters. We have 4 capital letters in the DNA, and those letters, they represent adenine. And then we have guanine, then we have thiamine, and then we have C, which is cytosina for C for cytosina. And then now we have RNA, ribonucleic acid, and over there we have capital A for adenine, then we have capital G for guanine, and then we have capital U for uracil, and then we have capital C for cytosina. We have capital C for cytosina. Yeah, I think that's what I was trying to figure out right there. Capital C for cytosina. So now, apart from that, we have sugar. Our DNA has sugar. Our DNA has sugar. I need sugar to survive in there. So our DNA has sugar for energy. So, and then the component in that sugar is ribose RNA. Ribose RNA or deoxyribose DNA. Those are the components that are contained in the nucleotides. Those are the components that are contained in the nucleotides. Apart from that, we have a phosphate group. So we're gonna learn more in our next topic. We still have more. So remember, we just covered up to the nitrogen base nucleotides. But now we're going to learn about some other elements that are in the system of our components of the nucleotides. So one of them is phosphate. We already discussed nitrogen. So next time we're going to discuss about phosphate. We are going to discuss about phosphates, and then we're going to look at the structure of the DNA, not the DNA, but of the chromosomes. We're going to look at the structure of the chromosomes. Remember, the chromosomes are those are the ones that are those strands that forms DNA. The chromosomes are those strands that are form the DNA. Remember, I told you they go up to 1.8 meters long. So now we're going to learn more about those in our next class. So we're going to learn about functions of DNA. We're going to learn about functions of DNA, and then we're going to learn about how DNA replicate itself, how DNA replicates himself. And now so let us just stop there for today. This topic can be fun if you start studying. If you don't stop yourself, you just can keep on going and going. So let's stop there for today, so we have time to digest what we have learned. Please follow this page at Paulstutorialservices.com. Continue to share, continue to click the follow button, continue to register for tuition, continue to give us your feedback. Please answer the survey. There's a survey at the page. There's a survey at Paulstutorialservices.com. Let us know what you think. Let us know what we think and how we can best serve you. Thank you. Please help us share. Till we meet again. Bye-bye.